Neurology and endocrinology. Neuro. Behavior. Genes, changing in static and then punctuated. Or gradualist, they code for proteins. Enzymes making neurotransmitters. Receptor for neurotransmitters. Endo. How reproductive status change the brain. Hormones affecting the nervous system. Two themes. Different ways the nervous and endocrine system can change its function over time. Individual differences, genetic influences, or environment. Dale's laws. 1. Neuron, axon, and the axon terminals. 2. Any given neuron is releasing only one kind of neurotransmitter. But, that's not the case. There are multiple neurotransmitters released by neurons. Exocytosis. Synapse with its vesicles filled with neurotransmitter comes the action potential vesicles move to the cell membrane dumps the neurotransmitter. So, people realized that neurons came with different colors of neurotransmitters in the vesicles. Even 3 types of neurotransmitters. When you get 2 neurotransmitters, in the same neuron, same axon terminal, and the same vesicle, they're structurally very different types of neurotransmitters. Like one made from a simple amino acid, another a complicated protein. So, they're going to have different mechanism and speeds of action. Where generally, one has a rapid and short-term effect, quickly decrementing, like, a little ionic excitability change. The other will have longer effect, like, change gene transcription in that neuron, or change structural stuff. Also, one will have receptors for it on the neuron itself. So, thanks to the different structures between the pairs of neurotransmitters you get different functions. Endo. Neuroendocrine axis. Pituitary-anterior pituitary-glandular cells releasing ACTH- you've got CRH, brain as an endocrine gland, the brain releasing hormones into the circulatory system, and out comes its pituitary hormone. CRH, the hypothalamic hormone at the base of the brain, released into the circulatory system stimulates cells dump ACTH into the circulation, usually to a distant gland. Hypothalamus release CRH transforms it into ACTH during stress. Or, the hypothalamus release vasopressin or oxytocin or norepinephrine or epinephrine, so collectively they stimulate the release of ACTH. What you have are stress signatures, different types of stressors will trigger different orchestrations of ACTH releasing neurons. Like low blood pressure or hypertension, release CRH and vasopressin. Low blood sugar, release CRH, epinephrine, and norepinephrine. Advantages. Depending on the orchestration, the shape of the ACTH secretory curve is different, like long, short duration, etc. Also, to know when to active different things on different parts of the body. Or, instead of releasing ACTH, they inhibit its release, corticotropin inhibiting factors. Like the peptide delta sleep-inducing factor which turn off your stress response while you sleep. How these systems work. Neurons. Depolarization sufficient to get to the axon hillock action potential ionic excitation all or none regenerating down the axon to the axon terminals. Dale's Law 1. An action potential started and release neurotransmitters from every axon terminal. Dale's Law 2. An action potential causes neurotransmitter to be released from every axon terminal. But, that's false. It's going to be the same neurotransmitter released from every axon terminal. Jerry Letvin. He sat in a dark abandoned warehouse for decades. And every decade he writes a paper. One paper from the 70s. Dale's Law number 1 was violated. You can get blockades on this, that would stop the action potential from propagating some of the branches of the axonal tree. So, it's not simple an action potential that dump neurotransmitters, there are other branches regulating another type of unexplored things. Hypothalamic hormone release pituitary hormones. So, you have specialized cells within the pituitary, that only secretes one type of hormone. Like, growth hormone equals somatotrophs. Prolactin equals lactotrophs. ACTH equals corticotrophs. FSH equals gonadotrophs. There's a mosaic all across the pituitary of all the different cell secreting types. So, you get local, little neighborhood effects. E.g. somatotrophs. One is in a FSH neighborhood. And other is in a prolactin neighborhood. Where the first one will release different amount of growth hormone than the other. Because there's all sorts of communication going on between the individual cells in the pituitary. So, the hypothalamus will release hormones to different parts of the pituitary where its cell targets are in different sorts of neighborhoods. Negative feedback. Neuron get excited release neurotransmitter into your synapse you clean it up after, remove the neurotransmitter from the synapse. An enzyme turns X into Y, it stops when there's a lot of Y, that inhibits X. E.g. autoreceptors. Where you not only will have receptors for neurotransmitter on the postsynaptic neuron on the other side of the synapse, but you'll have them on the neuron that's releasing the neurotransmitter, which is for bookkeeping. If the neurotransmitters bind their receptors to the next neuron, a certain number of them are going to bind to the autoreceptor. And there's a rule in the presynaptic neuron, that for every time one hits the autoreceptor, it means it will release 1000 copies of the neurotransmitter. And that's how it keeps track of the numbers, of feedback regulation. Often, one of the neurotransmitters will exclusively work on a presynaptic autoreceptor, in order to do the bookkeeping. Same with endocrine systems. Hormones get into the brain and change how you think, feel, behave. And also, letting the brain know how much hormones were in the circulation in order to do the negative feedback regulation. Glucocorticoid negative feedback regulation. The more glucocorticoids in bloodstream, the more likely they reach the threshold. So, the more of a negative feedback signal, the less CRH being secreted. Measuring the level of bloodstream is how most endocrine negative feedback works. But there's another domain. The brain is measuring the rate of change. On how many units of increase per second are you getting? The faster the rate of change, the less likely CRH is to get secreted. Mary Dahlman discovered this. Different parts of the nervous system that do each type. The very rapid rate of change, it's occurring at the pituitary, thus, it's not so much regulating CRH release, but ACTH release. Positive feedback. More of a hormone in the bloodstream equals more stimulate the system to do more. Autoregulation. Something going on at the receptor level. If someone is screaming you all the time, you downregulated your sensitivity, so you don't pay attention to him. And vice versa. So, in neuroendocrine, you change the amount of receptor for a neurotransmitter or hormone as a function of the levels in the bloodstream. And if you double the level of hormone, you cut the number of receptors in half to compensate, and vice versa. But if you don't compensate quite enough, or if you overshoot, like in depression, that's probably what's happening. What's wrong are levels of few different neurotransmitters, serotonin, dopamine, norepinephrine. That's why all the antidepressant drugs change the levels of these neurotransmitters in the bloodstream. But if you throw this type of drugs, SSRIs, they're changing levels of neurotransmitter within minutes to hours. Give it to somebody deeply depressed, and they don't start feeling better for days to weeks, it lagged it out. Because these drugs are not changing the levels of these neurotransmitters, they're changing the levels, they're eventually going to cause an autoregulatory change in the number of receptors. Or, also, causing an autoregulatory change in the autoreceptors. So, the amount of hormone and amount of neurotransmitter makes a difference, the number of receptors as well. And have big pathological changes in the levels of the messengers, the body attempts to regulate with autoregulatory changes. So, diseases are overshooting or undershooting. In endocrine, diabetes, glucose in the bloodstream. When you eat, you have nutrients in your bloodstream. Your pancreas detects the glucose in your bloodstream and secretes insulin, so insulin tells your fat cells to absorb the sugar and store it away. But the fact that your fat cells are already full, they stop listening to insulin, because they can't take up any more stuff and store it away. As a result, the pancreas secretes more and more insulin, but because they're full, the cells decrease the number of insulin receptors. Both do the same repeatedly, and at some point, your pancreas is working so hard to dump a bunch of insulin into the bloodstream that your body's paying no attention to, that you burn out the cells in your pancreas that make insulin. So, too much nutrients, thus too much insulin, the fat cells downregulate the receptors, both doing this over and over. Punchline. The amount of messenger is important, the sensitivity to the messenger is at least as important. You have as much capacity to regulate the neurons in biological systems, and thus has the potential for screwing up both. Pituitary is releasing hormones under the control of the brain. But you've got pituitary cells that only secrete prolactin, or only ACTH, etc. And, in any given cell in the pituitary, will respond to its specific hypothalamic signal by secreting whoever hormone it specializes in. Depending on what sort of neighborhood it's living in, depending on which other types of cells it's surrounded by. You end up with ways for regulation of the amount of secretion, by determining which way the hypothalamus turf the signals. The more glucocorticoids in the bloodstream, the more of a negative feedback signal. Also, with the other types of neurons, vasopressin, oxytocin, etc. By determining the level and the shape of glucocorticoids levels in the bloodstream, so there's feedback on all type of neurons. How receptors go about their business. Ligand, a neurotransmitter is a ligand for a neurotransmitter receptor. Or, a hormone is a ligand for a hormone receptor. The ligand is whatever the receptor normally binds. More of a ligand for a receptor, the more likely to trigger the compensatory downregulation. Another level of regulation. Receptors, their job is to bind their ligand to do something or other, and as a result the cell opens up some channel that allows a change in excitability in the neuron. 4 different receptors are made up of a number of different proteins of a complex of proteins. This is a receptor coded for by multiple genes. It means, we have the potential for variation, of ways of generating different versions of that same receptor, which will work a little bit better, or slower, etc. Another. A receptor made of 3 different proteins, and there's 4 different genes that make proteins that can construct one of those receptors. Like, 1-2-3, or 1-2-4, or 1-1-4, and so on. Another. Cells change the subunits on their receptors, degradation. Like, 1-2-3, something degrade the copies of 2, and replace it with 4. And change how well the receptor it's doing. E.g. glutamate receptors, for learning. They're not only increasing the number of copies of a certain type of glutamate receptor, but changing the subunit composition. Another. You've got an abnormal subunit, which produces a cell that's prone towards high levels of excitation. This is a congenital reason for causing epilepsy. All of these, regulating, mixing and matching the subunit stuff. All the flow of information from the dendrites to the axon hillock. But no single dendritic input is enough to trigger an action potential, you need to have enough summation, to reach the threshold of the axon hillock, to initiate the action potential, and the threshold could change over time. What does it mean, when an axon hillock's threshold is changed, those critical first channels that open up, you've changed the subunit composition. All of receptology and channels that open and close, where cells can change which are the pieces that make it up, and change the properties subtly. With hormone receptors, same deal. Steroid hormone receptors. 2 domains. 1 binds to the hormone. 2 binds to the promoter. But, steroid hormone receptors, have other proteins that they bind, cofactors. So, you get different arrays of cofactors in different cell type, which will again, activate different things. Receptors, a lot of them can bind more than one ligand. GABA, main inhibitory neurotransmitter in the brain. The GABA receptors binds to GABA, its ligand is GABA, found on the dendritic spines of GABA-responsive neurons, and the neuron is upstream of it, will release GABA in response to its action potential. And when it binds to GABA, it becomes less excitable, it's an inhibitory neurotransmitter. But GABA also binds to other things. 1 major tranquilizers, barbiturates, they bind to the GABA receptor, there's a side complex on it, so they make GABA more inhibitory, this is how anesthetics work. 2 minor tranquilizers benzodiazepine, Valium, and Librium, also have a side complex, they increase the force of GABA signaling. 3 progesterone also has his side complex, and also potentiates the effects of GABA. It has something to do with the mood shifts over the course of the reproductive cycle, like perimenstrual, because of a shortage of progesterone having minor tranquilizing effects. Circular neurons. Neuron, B-A. B releases an excitatory neurotransmitter, and thus, excites A, e.g., glutamate. And, GABA does inhibitory stuff, sends its projection to A. So now we have dendritic spines in A contain both receptors, glutamate and GABA. But this doesn't happen. Instead, GABA neurons never send a projection to A. They send the projection onto B. GABA is making the neuron less excitable, when B has started an action potential, that's coming down the axon, a GABA signal comes out that it silences it, it never gets to the end, and sumps the neurotransmitter. So, GABA is forming synapses not on the dendrites of B, it's forming a synapse on the presynaptic end of B. An axoaxonic synapse. Which it means, there have to be GABA receptors sitting at the presynaptic end. So, what does GABA neuron does to the excitability of A? It does nothing, it only alters the ability of neuron B to do something to A. GABA serves a neuromodulator role in the nervous system. Now, back to the major and minor tranquilizers, and progesterone. They don't inhibit the neuron, they modulate/potentiate the activity of GABA. With the same principle of modulation, in the endocrine system. E.g. the amount of ACTH coming out of the pituitary. You throw in some CRH on the pituitary, injected into circulation equals low ACTH. Throw in some CRH with vasopressin equals very high ACTH. Now, throw vasopressin equals no secretion. Because vasopressin potentiates and modulated the activity of CRH's activity. So, all the other releasers, vasopressin, oxytocin, epinephrine, norepinephrine, none of them are not direct releasers on their own, they all are modulating/potentiating CRH's effects. At the end, lots of individual variability, ways in which these systems are changing in response to experience. All the subunit changing, something happens, and you get rid of one subunit, replace it with another, and you've changed the excitability of the system. All of conditional if/then clauses.